Let's just dive right in. In today's Explainer, we're investigating cancer, and we're not just looking at it as a disease, but really as this incredibly complex biological hijacking of our own cellular machinery. It's a topic that touches pretty much all of us in some way, right? But today, we're going to look at it purely through the lens of biology. We'll be decoding the microscopic errors that lead to this condition and seeing exactly how a single microscopic cell can just completely rewrite its own rules.
To make sense of such a massive topic, we're going to follow a quick roadmap. We'll cover what cancer actually is, the 6 hallmarks of the condition, mutagens and oncogenes, how we profile blood cancers, and finally, how modern treatments actually work.
So, let's start at the very beginning. What exactly is cancer?
The absolute most crucial thing to understand here is that cancer is, fundamentally, a complete loss of control. Normally, our body cells are under incredibly strict regulation. They differentiate, they divide, and they eventually die in this highly orchestrated, orderly sequence. Cancer is basically an abnormal cell that just absolutely refuses to follow that sequence. And it's kind of wild, but the word cancer actually comes from a Latin word meaning crab. Early observers thought the way these abnormal cells migrate, adhere to things, and cause pain looked a whole lot like a crab grabbing onto tissues.
Now, we really need to highlight the stark contrast between two very different types of cellular growth. On one hand, you've got benign tumors. These guys are slow-growing, highly localized, and they're neatly contained inside a protective capsule. They look pretty much like the normal tissue they came from and are rarely life-threatening. But on the flip side, we have malignant neoplasms. These are the rule breakers. They're fast-growing, poorly differentiated, and they totally lack that capsule. Because they aren't contained, they actively invade surrounding normal tissues, and, most dangerously, they metastasize. That means they literally hijack the bloodstream or lymphatic system to travel and set up camp, forming secondary tumors in completely different parts of the body.
All right, moving on to part 2. Let's talk about the 6 hallmarks. Back in the year 2000, 2 scientists named Hanahan and Weinberg laid out something really profound. They identified 6 major hallmarks that show exactly how a normal cell transforms into a self-sustaining rogue entity. Think of it like a dark checklist of superpowers that a cell actually has to acquire before it can become a successful cancer.
Let's break down what these 6 traits actually mean for the body, because it's fascinating. First off, they produce their own growth signals. They basically become entirely self-sufficient. Second, they go completely deaf to the body's anti-growth stop signals. They just ignore them. Third, they actively evade apoptosis, which is the body's natural programmed self-destruct sequence for damaged cells. Fourth, they achieve limitless replicative potential, meaning they can divide infinitely, no built-in expiration date. Fifth, they sustain angiogenesis. This is crazy. They literally force your body to build brand new blood vessels just to feed their own massive oxygen demand. And finally, as we touched on, they acquire the ability to invade tissues and metastasize. By checking off these 6 traits, a tumor entirely manipulates its microenvironment.
Which brings us to our third section, mutagens and oncogenes.
So, we really have to ask ourselves, what exactly triggers a perfectly healthy, totally normal cell to just suddenly rewrite its own biological programming? Well, the answer lies in the ultimate critical macromolecule, our DNA. Carcinogenesis, which is just the fancy term for the molecular process of creating a cancer cell, always starts with damage to our genetic code.
Think of a cell's division cycle kind of like driving a car. Our cells have these things called proto-oncogenes that act exactly like the gas pedal, promoting normal, healthy cell division and growth. On the flip side, we have tumor suppressor genes acting as the brakes, stopping growth when something looks wrong. But when a mutation occurs, that delicate balance is completely shattered. It's like the gas pedal gets glued to the floor, transforming that normal proto-oncogene into an overactive oncogene. And at the exact same time, the brakes completely give out.
And how does that happen? Well, usually through exposure to mutagens. We're talking about things like physical radiation, carcinogenic viruses like HPV, or certain chemical carcinogens. These things can literally flip a genetic switch. A proto-oncogene that should be in the off position is forcefully activated into an oncogene in the on position. And just like that, this single genetic change gives birth to an actively dividing abnormal tumor cell.
Now, knowing that chemicals out in the world can cause these mutations, how do we catch them before they harm us? Enter the Ames test. This is a genuinely brilliant, incredibly cost-effective screening tool. Here's how it works. Scientists take mutant salmonella bacteria that literally cannot grow without being fed a specific amino acid. Then they expose those bacteria to a suspect chemical.
If the bacteria suddenly mutate back and regain the ability to grow completely on their own, bingo, you've just proved that chemical is a mutagen, and it's very likely a human carcinogen that we need to keep far, far away from our food and environment.
Let's shift gears a bit for Section 4 and look at profiling blood cancers. When we talk about systemic blood cancers, there are three major case profiles we really need to understand. First up is leukemia. This one begins deep in the bone marrow, causing a massive, rapid spike in immature white blood cells, called blasts, that end up completely crowding out your healthy cells. Then you have lymphoma, which targets the lymphatic system. So we're talking about your lymph nodes and spleen, and it specifically messes with your B or T lymphocytes. Finally, there's myeloma. This one specifically attacks plasma cells, which are the mature B cells whose normal job is making our antibodies.
If we zoom in on lymphoma for a second, you'll see it breaks down into two main categories. The much rarer Hodgkin type is definitely marked by the presence of these large malignant things called Reed-Sternberg cells, and it usually spreads in a very orderly, contiguous way, literally moving from one lymph node group right to the next door neighbor. But on the other side is non-Hodgkin lymphoma, which accounts for about 90% of all cases. It totally lacks those Reed-Sternberg cells, and it spreads in a way more unpredictable manner, often popping up in extranodal sites like the skin or even the bone marrow.
And if you want to understand the truly devastating full-body effects of myeloma, medical students use the acronym CRAB, C-R-A-B, because these myeloma cells pump out abnormal antibodies. They just wreak havoc everywhere. The C stands for elevated calcium, which happens as bones are quite literally destroyed. The R is for renal or kidney failure, because damaging proteins get deposited there. The A stands for anemia, since the bone marrow gets completely hijacked. And the B is for severe bone lesions and fractures. It perfectly and tragically highlights how a single cellular error can cascade and crash the entire system.
All right, let's move to our final section, how treatments actually work. So, traditional chemotherapy essentially exploits one primary biological fact. Cancer cells divide way, way faster than normal cells. These cytotoxic drugs act like molecular wrecking balls. They shatter the cancer cell's ability to replicate, sometimes by cross-linking its DNA or by totally messing with the microscopic scaffolding the cell needs to divide. The ultimate goal is forcing the cell into apoptosis, or self-destruction. But here's the catch, and we all know it. These drugs are blunt instruments. They also hit perfectly healthy, fast-growing cells, like our hair follicles and the lining of our stomach, which is exactly what causes those severe, well-known side effects.
But the next generation of treatments, that's where things get incredibly exciting. We're talking about highly targeted therapies. Take a mutant RAS protein, for example. In a healthy cell, RAS is just a normal switch. It turns on to signal growth and turns off when it's done. But a mutation leaves it permanently jammed in the on state. Instead of just indiscriminately carpet bombing the whole body, modern targeted therapies act like a sniper. They are chemically designed to specifically locate and intercept broken pathways exactly like this one.
So, instead of those blunt tools, oncology is increasingly relying on these highly specific interventions. For instance, we now use cytostatic tyrosine kinase inhibitors. I know, big words, but basically they starve tumors by blocking the exact enzymes the cancer desperately needs to signal growth. We're also using monoclonal antibodies. These are immunotherapy drugs, literally designed to paint a target on cancer cells, basically waking up the patient's own immune system so it can swoop in and destroy the rogue invaders. We are quite literally retraining the human body to defend itself.
Understanding this incredibly precise sequence of molecular transformation, literally tracing the path from one single DNA mutation all the way to a fully metastasized tumor, has been the master key for modern science. It's allowed the entire field of oncology to evolve. We're no longer just treating cancer as some mysterious, unavoidable illness. We are actively intercepting it as a mechanical failure, using incredibly precise biological solutions.
I really hope this explainer has helped demystify the intense biology behind cancer for you. By breaking down the hallmarks, those tricky genetic switches, and the ingenious ways modern medicine is fighting back, it becomes clear that cancer is, at its core, just an error in our complex cellular system. So I'll leave you with this thought to chew on. As these targeted therapies continue to completely rewrite the rules of the cellular battlefield, are we finally on the verge of learning how to permanently reprogram the ultimate error in our biological system? Thanks so much for joining me today, and keep questioning how the amazing world inside us truly works.